Welcome to Chapter 9, Topic 1. This is our second to last chapter in Unit 2. And in this chapter, we're going to talk about all the genes and information we've been talking about in chapters 5 through 8. We're going to talk about how those genes have come to evolve to be the genes that we know they are today. So in Topic 1, we're just going to talk about how genes in the genome evolve, and then Topic 2 will move into a little bit more discussion on this. So here's our topic outline for today. We're going to talk about how we generate genetic variation. So we're going to talk about mutations within a gene, mutations within a regulatory region, gene duplication, exon shuffling, and horizontal gene transfer. As always, here is your topic objectives for this topic, as well as you'll have your own set for the next topic. So you'll have two sets for this, for Chapter 9. Make sure you understand the differences between these. These are really big objectives that you'll need to be able to have mastered before our next test. As always, if you have any questions about any of these, please just let me know and we'll talk about them in class. All right, so hopefully you remember from your earlier biology classes that evolution is driven by change over time, and that is based in genetic variation. We have to have genetic variation in order for there to be change. And then there's environmental pressures that lead to that. And so that's where you see all of the different genetic variations that we see on this planet. And you can even see that within the human population alone. Even within your family, you should see different genetic traits between them all. And so it's really important to understand that genetic variation is very important to biology and to the world around us and to continuing life in the future. So let's talk about these genetic variations. As I already mentioned, these are the ones we're going to talk about this topic, but let's go through them again. So the first one we're going to do is mutation within a gene. We've talked about these ones. This is where a mutation will occur within a gene. This is like sickle cell, for instance, that we've talked about before. Mutation within the regulatory region is where the regulatory genes, remember we talked about those before, this is when the regulatory region is turned off when it should be turned on, and so now this function, or now this gene is not going to be expressed. Or it should be turned off and now it's turned on. Gene duplication is where we literally duplicate the gene within the genome. Exon shuffling will change the way that that gene is read, packaged, processed, etc. And then we have horizontal gene transfer, which is conjugation in bacteria. It's where an organism will directly pass genes to another organism. And we'll talk about that at the end of this topic. So the first thing we have to get straight is that as far as genetic variation goes, the only important mutations, the only important changes, they have to occur in germ cells. And germ cells, so what are germ cells? We've mentioned this before, but let's go through the definitions again. A germ cell is a cell type in diploid organisms that carry only one set of chromosomes and is specialized for sexual reproduction. That in humans is eggs and sperm. The somatic cells are basically any other cell within the organism. And this is not the germline or the germline precursors, so it's just anything else. It's your skin cells, your hair cells, your eye cells. Now, there can be mutations that occur in your somatic cells if they occur early enough. Well, they're not somatic cells at that point, they're still stem cells. If they occur early enough in the zygote, that can then be passed down into germ cells. But as far as passing from parent to offspring, they have to occur in the germ cell line in order to be passed down. You can see that on this image here. Make sure you understand this graphic. This is a very important graphic, just like the one we talked about before with the different regulation steps. This one's another one of those really important graphics to make sure you understand how this works. So make sure you understand that in order for variation to be passed on, it has to occur in the germ cell line. So let's talk about some examples. Point mutations within genes, or changes within the genes. Now, a lot of point mutations are neutral mutations, which means they don't change at all. There's no well, they change, but there's no visible result. We can't tell without sequencing the gene if there's anything different between that gene and a normal gene. There are also ones that could cause all sorts of disruption, all the way from small changes to lethal changes. And one of the best examples is sickle cell. Remember, sickle cell is caused by one amino acid change within the sequence, and it changes the entire shape of that red cell or red blood cell. So make sure you understand that how these point mutations can range. They're not always dramatic changes. Sometimes we don't even notice them. Now, these mutations can happen pretty quickly. And this is one of those things we talk about, how our error rate is 1 in 10 to the 9th cells. Now, that seems really high, but when we start talking about lots and lots of cells, for example, E. coli, which replicates really quickly, if we were to put it into a solution, so if we had the example of this experiment here is we have a mutant E. coli that can't produce histidine. So we put it in a media that contains histidine so that it can grow, because otherwise it wouldn't be able to. And then we culture it out pretty quickly. We're going to find mutants that are now capable of producing histidine. And so that shows that these mutations can occur pretty frequently within a population. But remember, that's just because we're talking about massive amounts of generations of E. coli really quickly. It's not necessarily saying that tomorrow someone is going to pop up with wings. It's simply just saying is that these mutations can happen within generations pretty quickly. So another important aspect is regulatory genes. Now, in fact, I would almost argue that regulatory mutations can be more harmful than point mutations within the gene. And this is because regulatory proteins are really important, and as we talked about, they're important for differentiation of cell types. They're important for regulation of the cell. And so we need to make sure that we understand that these are kind of a big deal. And they can be even harder to detect because we have a hard time determining the results, because remember, regulatory genes can control more than one gene. They can change the outcomes. They can just make small changes to how much is expressed versus not just complete turning on and off. And they can occur just as quickly as point mutations within genes. And as I said, they can silence or they can activate, or they can have a variation on it. And one example of this is lactose intolerance. And humans are one of the few organisms that can consume milk as adults, and that's because we produce an enzyme known as lactase. However, there's a growing population of people who can't produce lactase, and so they're lactose intolerant. And you can see an image of that on the slide here. And so you can just see that that's just one example of how point mutations within regulatory genes can affect normal functioning of the cell. All right, so let's talk about gene duplication now. Gene duplication occurs through homologous recombination. And basically, instead of aligning properly like it's supposed to, like we talked about in chapter 8, this point, what's going to happen is there's going to be an unequal crossover. And so now one chromosome has a whole bunch of genes on it when one chromosome doesn't. And so this essentially puts two copies together, and the gene on one chromosome has 2 copies, one chromosome has 0 copies. And then you can see how that works here. And then this can cause errors to be made in how things are read, because if the genes are in different places, they can't be read the same way. And so it's really important to understand how that works. Now let's talk about how this is actually beneficial. And here's an example of gene duplication. In gene duplication, in this example, we see the development of our 4-hemoglobin chain molecule that we use to carry blood on our red blood cells. And it started out as just a single globin that bound to one oxygen molecule. And over the course of time, there was enough copying of this gene that created the 4-molecule structure that we know today. And so this is how these gene duplications have actually had a positive impact on, for us, for oxygen carrying, but there's also a lot of different other aspects of it too. Now, just because we talk about just a single gene doesn't mean that the whole genome can't be duplicated, and it can. The whole genome can be duplicated. Now, in humans, we've seen that this is not exactly something that could happen pretty regularly and be compatible with life, since we know that even a single extra chromosome on chromosome number 21 creates some pretty dramatic effects in with the result of Down syndrome. So that's why it's unlikely that we're ever going to become triploidy, because of the fact that even a small addition of our chromosomes has it creates more problems than good. But there are some examples here. For instance, you can see the frogs where they have an entire genome duplication and they're able to survive. Now here's another thing. We've talked about mRNA processing, where we remove exons, or how some exons are removed, some are not. It all creates different things. Well, if the exons are placed in the wrong place through homologous recombination, we're also going to get miscopied, mistranscribed. There you go, mistranscribed mRNAs. And so it's really important these exons don't get moved around, except for in places where it actually helps. But for the most part, we try to make sure our bodies try to make sure they stay in the right place so that they don't get misaligned. Because as you can see here, this could create an entirely different mRNA transcript as opposed to the normal transcript that we're supposed to have. And this may be something that you guys are seeing in some of your diseases that you're working on this semester, that this is where some of the problems have come from. So as I said, with the exon shuffling, well, that was exon duplication. Now we're going to talk about exon shuffling. And this is where they move around. And it's similar, but it's moving, not just copying. And the domain shifts are due to a diverse group of products, or the domain shifts can cause a diverse group of products. So what happens is by moving it around, now the domains of the proteins are different. And this can cause a lot of different things. You can see examples here on this slide of what this works for, of how this exon shuffling works. And this is why exons and exon splicing and all of that is so important, because it can create a variety of products of protein from a single mRNA based on what exons are removed and which exons are not. And so if the exons are shuffled around, they can't be read quite as easily. And this explains why one gene can create so many different proteins. So mobile genetic elements and genetic variation causes this problem too. In this example, you can see how the mobile genetic elements, by moving around, have dramatically changed the shape of this insect on the slide. And this is exactly what happens with these mobile genetic elements, is that they will move in, they will change the way the mRNA is going to be transcribed and create different outcomes within the organism. So remember, mobile genetic elements can create just as much genetic variation. We've already talked about an example this semester with blood clotting disorders and how mobile genetic elements can create that. The last item on our list for creating genetic variation is horizontal gene transfer. Now, it can occur in eukaryotes, but it almost always occurs in prokaryotes, and that's it. And it's called conjugation in prokaryotes. And this is where a gene is transferred specifically from one bacteria to another bacteria. And this is how they pass on antibiotic resistance or other important genes like that. And so don't worry too much about it for eukaryotes, but do understand that they do pass on entire genes through this process, usually using plasmids from one bacterial cell to the other. And this is how we can spread it. So this is the end of topic 1. Make sure you have a good handle on how all of these different processes can create genetic variation. And then when you're ready, feel free to move on to topic number 2.